So, am I audible? Audible? Yeah. Okay, ji. So, if we talk about certain historical aspects of the cell biology, cytology. So, I'm just writing the name of scientist. Robert Hooke. He is credited for which one? Discovery of cell. So, he observed a honeycomb-like a structure present in cork cell. In cork. And these honeycomb-like structure who present in the cork, he called as what? Cell. Definitely of oak tree. So cork is living or dead? Cork is living or dead? It is dead, na? So he just observed the which cell? Dead cell. Is it clear? So A.V. Leeuwenhoek. He is credited for which one? He first observed the living cell. So if someone going to ask a question, first living cell is observed by, answer will go to Leeuwenhoek. If someone asks a question, cell is discovered by, answer goes to whom? Robert Hooke. So keep these things in mind. If they are specifically asking living cell, then you have to give the name of which one? Leeuwenhoek. Clear? So these are the things where some confusion occurred in students' mind, so I clarified it. Robert Hooke seen the dead cell, right? Now I am telling you about another name, Robert Brown. He is associated with discovery of nucleus. Right? Fontana, associated with discovery of nucleolus. Nucleolus. Knoll and Ruska, associated with discovery of electron microscope. So these are the few scientists name you keep in your mind. Right? Robert Brown, Fontana, Knoll and Ruska, A.V. Leeuwenhoek, and Robert Hooke. Now scientist based, not much questions is going to ask, so definitely we are not going to tell you a long list of the scientists. So these are the name you keep in your mind associated with the name of scientist in the cell structure chapter, right? Later on, when plasma membrane we will discuss, we will talk about some more scientist name. Getting the idea? Is it clear in the mind? Right. So now we are coming on cell theory. Cell theory. Right. So if we talk about cell theory, it is given by Sleeden and Swan. Sleeden and Swan. Sleeden was a botanist. He belongs to which country? German botanist. And if you talk about a Swan, he was a zoologist and British zoologist, right? Okay, ji? Right? So, if we talk about Sweden, what was the observation of Sweden? Sweden observed different plant species, right? And conclude that all plants are made up of cell. All plants are made up of cell. These are the observation of whom? Sweden. And what was the observation? Observation is all plants are made up of cell, which form tissue of the plant. So he told each and every plant have cell, and these cell when aggregate together, they are forming a what? Tissue. This is the observation of Sweden. Is it clear? Now we are going to talk about what was the views of the British zoologist Swan. What was the views of which one? What was the view of Swan? So Swan, he observed the different animal cell and concluded that the outermost boundary in animal cell is a thin membrane. Getting the things or not? He observed the different animal cell and observed what? The outermost boundary is made up of which one? Thin membrane. Nowadays it is called plasma membrane. Nowadays it is called plasma membrane. He don't say about plasma membrane. He say first thing what I not wrote here, what he observed, the different animal, not better to write cell, different animal. So what the conclusion is there? And conclude that, you write it in second conclusion. The outermost boundary in animal cell is made up of thin membrane, and first conclusion was what? All animals are made up of cell. So all plant are made up of cell, told by whom? Sweden. All animals are also made up of cell, told by whom? Swan. And what Swan told further? Ki the outermost boundary in an animal cell is a which one? Thin membrane. And nowadays we call this membrane as a plasma membrane. Getting the idea or not? He don't use the word plasma membrane. Keep this line in mind. He simply told that time plasma membrane name was not there. He told there was a thin membrane present in the outermost boundary. So first observation, animals are made up of cell. Second observation, outermost boundary of the animal cell is a thin membrane. And he also told another thing. I am telling the third observation. That is what? Presence of cell wall is unique feature of plant cell. So cell wall present in a plant cell, that line is told by whom? Sleeden or Swan? Swan. Keep this line in mind. Many times students do the mistake in flow when they not revise it repeatedly. What flow? Botanist, Sleeden. So they relate the plant cell wall with which one? Botany. Getting the idea or not? It is told by the zoologist. That is what? Presence of cell wall is a feature, unique feature of which one? Plant cell. Is it clear or not? So how many things told by Swan, which is very important to know? Three things. First, animal are made up of cell. Second, animal cell have a thin membrane, outer membrane. Third, plant cell have a unique feature, that is what? Presence of cell wall. Is it? Further, he also compiled the cell theory. Getting the things or not? And finally, what he told? All organisms are made up of cell. In all organism, who is coming? Plant and animal both, made up of cell and its product. All organisms are made up of cell and its product. Getting the idea? These are the line compiled by whom? Swan. So if anybody going to ask a question, who hypothesized the cell theory? What the answer you will give? Who hypothesized the cell theory? Who gave the hypothesis of cell theory? Swan. Question going to ask, who cell theory is given by? What the answer will you give? Both Sleeden and Swan. Getting the thing? Cell theory is given by Sleeden and Swan both, and Swan hypothesize the cell theory as what he told? All organisms are made up of cell and its product. Look, in beginning, I already told you. What in beginning I already told? Have a look here. In very beginning, I already told, that is what? Cell theory is given by Sleeden and Swan. Right? In very beginning, the first line I wrote. So not make a confusion. There are two key points where student confused. Beside of that, it is damaging. One, who given the cell theory, either Swan or Sleeden, and both Sleeden and Swan. Who hypothesized the idea, compiled the idea of both? Swan. Is it clear? Okay, ji, done, over. Now we are moving ahead. Now we are going to deal about, later on we are going to deal, who modified the cell theory, who have given the modern cell theory. So then we will say about the name of which one? Rudolf Virchow. Getting the idea or not? So actually, Sleeden and Swan don't have idea about how a new cell evolve. They don't know about it. How a new cell evolve, they don't know about this thing. Able to understand or not? And this is told by which one? Rudolf Virchow. What told by Rudolf Virchow? Tell me. What told by Rudolf Virchow? Omnis cellula e cellula. Right. Omnis cellula e cellula. Right. What are the English translation of this omni cellula e cellula? That is what? All cell arise from pre-existing cell. pre-existing cell. So how the new cell evolved? How the new cell will evolve? First time these things told by Rudolf Virchow. That idea don't have in the mind of Sleeden and Swan. He simply observed the cell and told they are made up of plant and animal, and they told they are made up of cell and their product. But how the new cell is arising, that is told by which one? Rudolf Virchow. And these gentlemen, he modified the cell theory. He modified the cell theory. And now the modified cell theory as what? As first line he added here own version, that is what? Sleeden and Swan version. First point, what are the first point? All organisms are made up of cell and their product. The first line he added what is told by Sleeden and Swan, and second line he used which one? Added what he told this line. Okay, that is what? All cell arise from pre-existing cell. Pre-existing cell. Getting the things? So you can say it as what? Modern cell theory. Modern cell theory. Getting the idea or not? Is it clear? Is it clear? Right? It is over. So what one line he added extra? That is what? All cell theory arise from pre-existing cell. Is it clear? Right. Now we are moving ahead. Now we are moving ahead. Now we are moving toward which one? Type of cells. So if we talk about cell, there are two different type of cells we are reporting. One is what? Prokaryotic cell. Prokaryotic cell. Another is what? Basically eukaryotic cell. Since 7th class you are studying about it. What are the basic difference between prokaryotic cell and eukaryotic cell? So prokaryotic cell, you just keep in mind, pro means before and karyon means nucleus. So that cell who came in nucleus came in existence without evolution of nucleus, without presence of nucleus. They came before nucleus. It is called prokaryotic cells. So what you will say in the prokaryotic cell? Well-defined nucleus is absent. Well-defined nucleus is absent in prokaryotic cell. And of course, here it is present. Here, well-defined nucleus. Eu means true. And karyon means nucleus. Cell with true nucleus. Getting the idea or not? Is present in which one? Eukaryotic cell. Is it clear? Right? Now, if nucleus is not present in prokaryotic cell, then who is present? Who do the job of the nucleus here? So you can say in prokaryotic cell, incipient nucleus is present. Incipient nucleus is present. Is it clear? Incipient nucleus I am writing. In prokaryote, incipient nucleus is present. Now question arises, what is incipient nucleus? So you can say these incipient nucleus is also called nucleoid. This is also called nucleoid. Able to understand? Clear in the mind? Incipient nucleus or nucleoid is present. These nucleoid you can also say as what? Genophore somehow. Genophore is present. Genophore is present. Getting the things? Or you can say about which one? Single. Or you can say single, circular, double-stranded DNA is present without histone protein. Getting the things? And these DNA are naked. These DNA are naked. Naked means not wrapped with, which one? Histone protein. Histone protein. So we are saying naked DNA. I am just drawing a cell. Suppose that this is a prokaryotic cell. This is a what? Prokaryotic cell. And in this prokaryotic cell, who is there? Here it is the DNA is present like this. Getting the things or not? So it is what basically single circular, you make circular better, single circular DNA. Here it is single circular DNA. You can say single circular DNA as nucleoid, as incipient nucleus, as genophore. Able to understand or not? Getting the things or not? Is it clear? And of course, who is absent? Well-defined nucleus is absent. Nuclear membrane is absent. Nuclear membrane is not there. Nuclear membrane is absent. So this DNA is present in which one? Cytoplasm. This single circular DNA present in cytoplasm. Present in cytoplasm. Is it clear? Is it clear in the mind or not? So we can say true chromosome not present. True chromosome not present. Getting the idea? Many times people ask a question ki how many chromosomes are present in a prokaryotic cell. In that case, you give the answer how much? 1. You can't say 0. Ki sir, true chromosome not present. Means they are chromosome in the sense they are writing about which one? These single circular DNA. They are not wrapped with histone protein. They are wrapped with which one somehow? Non-histone protein. Getting the idea? Is it clear in the mind? So, did you got about the nucleoid or not? Incipient nucleus or not? Tell me first. What I told about nucleoid or incipient nucleus? Well-defined nucleus is not there. Nuclear membrane is not there. If nuclear membrane is not there, there is no special compartmentalization. Simply DNA is there. These DNA are double-stranded. How many? Single DNA, naked DNA, not wrapped with which one? Histone protein. And these DNA are present in which one? Cytoplasm. Able to understand? And this things is also called what? Nucleoid. This things is also called what? Incipient nucleus. These things is also we can say like what? Genophore. Is it clear? It is all right. So what is present in the prokaryotic cell done? Now first we are going to write about this side. Here you will find in eukaryotic cell what you will find? Well-defined nucleus is present. Defined nucleus is present in case of which one? Eukaryotic cell. Is it done? Next we are moving. Next character I am moving. One I already discussed about the what is incipient nucleus. So second things we are going to see. In a prokaryotic cell, no compartmentalization. Present in cell. No compartmentalization present in cell. Cell don't have many compartments. If you talk about a prokaryotic cell, look here, what you will find? These are the cell membrane, innermost layer I am just telling, assume. So here you will find simply cytoplasm is there. Inside the prokaryotic cell, further compartment you will not find in the cytoplasm. But in eukaryotic cell, who is present? Compartmentalization present. Compartmentalization present. What is in compartmentalization here? See, here it is cytoplasm. One compartment is nucleus, another compartment is mitochondria, another compartment is which one? Chloroplast, or you can say Golgi complex, endoplasmic reticulum. So what you will find? Cell is divided into many small cytoplasm is divided into many small compartment. Are you getting the idea? And these compartmentalization is made by which one? Of course, membrane bound organelles. So who is forming this compartment? Compartmentalization formed by membrane bound organelle. Compartmentalization formed by membrane bounded organelle. Membrane bounded organelle. Getting the things or not? Is it clear? Right? So compartmentalization not present. Now another differences we are going to see about the prokaryotic and eukaryotic cell is what? All double or single. Membrane bounded organelle absent. Membrane bounded structure or organelle is absent. Here it is present. Membrane bounded organelles, either it is double membrane bounded or single membrane bounded, it is present in eukaryotic cell. So here who is the membrane bounded organelle present? Example, you can say mitochondria, you can say plastid, you can say nucleus. Getting the things or not? You can say about ER, Golgi body, etc. Lysosome, vacuole, you can say lysosome, vacuole, etc. are present. These all are present. Here these all are absent. Are you getting the idea or not? Is it clear? Right? So membrane bounded organelles are absent in which one? Prokaryotic cell, and present in which one? Eukaryotic cell. Out of these much, you better know these three. These three have how many membrane? Mitochondria, plastid, nucleus is surrounded by double membrane. DM, double membrane bounded organelle. And if you talk about ER, Golgi complex, lysosome, vacuole, they are surrounded by how many membrane? Single membrane. SM, single membrane. Able to understand? Now we are moving ahead. Now we are going to deal some more differences between which one? Prokaryotic cell and eukaryotic cell. Have a look here. So here I am writing ribosome is present. Ribosome is present in both prokaryotic and eukaryotic cell. Here also ribosome is present. Why ribosome is present? Because ribosome is a non-membrane bounded cell organelle. In ribosome, no membranes is there. Able to understand or not? Clear? Ribosome is a non-membranous structure. You can say it is non-membranous. Non-membranous. It is not surrounded by any membrane. And it is universal in every cell type. Na? Prokaryotic cell as well as eukaryotic. Is it clear in the mind? But nature of ribosome, in prokaryotic cell, ribosome is of 70S type. Ribosome is which type? 70S type. What is this S? We will deal during ribosome. And here ribosome, which kind of ribosome is present in eukaryotic cell? Ribosome is both 70S and 80S type. 70S ribosome present in which one? Mitochondria of eukaryotic cell, mitochondria and plastid, getting the idea, and 80S present in which one? Cytoplasm. So in overall cell, both 70S and 80S are there. Is it clear or not? So ribosome is both 70S and 80S. Is it clear? All right. 70S in cell organelle like what? Mitochondria, plastid, these two cell organelle also have ribosome. And that is the reason ribosome is also called organelle within a organelle. It is the only cell organelle present in another cell organelle. Is it clear? Right? So about ribosome we have discussed in a which one? Which type of ribosome is there in prokaryotic and eukaryotic cell. Now what we are going to deal? Now we are going to deal about cytoskeleton absent. Cytoskeleton absent in prokaryotic cell, not present in prokaryotic cell. And here it is of course present in eukaryotic cell. You know na cytoskeleton. Cytoskeleton is what? Can you tell the example? Microtubule, microfilament, intermediate filament. So these cytoskeletal structure are absent in prokaryotic cell and it is present in which one? Eukaryotic cell. Getting the things or not? Now if we talk about some more differences, so you can say cytoplasmic streaming. Absent. Here it is present. So actually what's happened? Cytoplasm are in which one? Rotatory movement. Able to understand? It is in rotatory movement. We will deal during transport. So these cytoplasm rotating always, showing a streaming. So it is called cytoplasmic streaming. This is present in eukaryotic cell and these cytoplasmic streaming is what basically absent in eukaryotic cells. Present in eukaryotic cell and absent in prokaryotic cell. So that is the reason I wrote cytoplasmic streaming is what? Absent here, present here. Able to understand? So these are the some of the basic differences we have seen about, and you know it from 7th, 8th class, between which one? Prokaryotic cell and eukaryotic cell. If you want to write more, there is many possibilities there, na? In prokaryotic cell, who is more stable? DNA is more stable. In eukaryotic cell, DNA is less stable. You can write like if you wish. So here, prokaryotic cell, DNA is more stable. And in eukaryotic cell, DNA less stable. Use your logic, why? Getting the things. Use your logic, why? We will deal it in which one? Next chapter. Able to understand? Ki why more stable DNA present in, why more stable DNA present in which one? Prokaryote, and less stable DNA present in eukaryote. Is there any significance of this is there? We will deal in the next chapter. Otherwise, here a lot of things are required to deal today. Able to understand or not? Right? So meanwhile, you just use your brain why I wrote this word. I will give later on explanation when required topic came. Getting the things or not? So next we are moving. Now we are going to see about the differences between plant cell and animal cell. Plant cell. And here we are writing about which one? Animal cell. Can you tell me few differences between plant cell and animal cell? You just note it out here. Who is present? Cell wall present. Here absent. If you talk about plant cell, plastid present. Plastid present. Here absent. Okay. right? If you talk about plant cell, centriole usually absent. Usually absent. And where it is absent? In which one? Higher plant. Getting the things? Centriole may present in some lower plant. Getting the idea? So here centriole present. Got the idea? In some lower plant also centriole is present. So don't say abruptly, it is what? Absent. It absent in higher plant. But it may present in which one? Some lower plant. If you talk about large central vacuole present in plant cell, large central vacuole absent in animal cell. Getting the idea? So if you talk about in plant cell, nucleus and cytoplasm is peripheral in position. Peripheral in position. And here nucleus centrally located. Centrally located. Nucleus is centrally located. Getting the things or not? Okay, ji. So here we can say when nucleus and cytoplasm is peripheral in position, that is, it is called primordial utricle. Primordial utricle present. Here it is absent. What is this primordial utricle? This peripheral cytoplasm. You just look, this is a plant cell. So in plant cell, the largest area is occupied by what? Vacuole. So here vacuole is there. So these are the cytoplasm. Na? Cytoplasm is thin and peripheral in location. But in animal cell, what you will report? In animal cell, you will find nucleus is centrally located and cytoplasm is occupied the major portions. Able to understand or not? Getting the idea? Nucleus is centrally located. Here, if you talk about the nucleus, it is located in periphery. So this peripheral cytoplasm is called what basically? Primordial utricle. It is large vacuole. It is large vacuole, central vacuole. Getting the idea? So primordial utricle present, primordial utricle absent. If you talk about some more differences in plant and cell and animal cell, what you can say about less number of mitochondria? Less number of mitochondria. Here you will find more number of mitochondria. More number of mitochondria. Able to understand? Getting the idea? So these are the, anything we missed, right? Which are very important? You just tell me. Almost we just talked about the which one? Difference between which one? Plant cell and animal cell. Able to understand? Clear? It is all right? Done? So plant cell, animal cell is also over. Now we are moving ahead. Give your attention. Should I change it? NCERT wrote it in beginning. Now I am asking a question to you people. Now I am asking a question. Please answer it. Tell me 2-3 similarity between prokaryotic and eukaryotic cell. One similarity, ribosome. Very good. Another similarity, both have plasma membrane. Another similarity, both have cytoplasm. So similarity is what? Pro and eu. In short, I am writing. Similarity. Similar nature. What are the similarity? Presence of cytoplasm. in both. Here also present. Here presence of plasma membrane. You can say plasma membrane present. Here also present. Third, you can say about which one? Ribosome. Present in here also, here also. Next, you can say genetic material. Is DNA here also, and here genetic material is DNA in this one also. So in both genetic material is same, DNA. Both have cell membrane, right? Cell membrane is a universal structure. Cytoplasm is a universal structure. Any cell is there, cell membrane will be there. Any cell is there, cytoplasm will be there. Getting the idea or not? Is it clear in the mind? Okay, ji? All right. So that is the reason NCERT wrote, na? That is what? Cytoplasm is the main arena of cellular activity. So what are there to make in the cell to make them living? These all activity occur in which one? Cytoplasm. Is it clear? It is all right? Right. So cytoplasm is the main arena of which one? Cellular activity. You got the idea? Understand the idea? Because cytoplasm, without cytoplasm, you can't imagine about the existence of the cell. Is it clear? All right. Now we are moving on. Size of the cell. You better know. So first we are going to talk about mycoplasma. You have to tell me the cell size. Then I am writing bacterial cell. What data given in NCERT, you please note down. Third, I am writing about which one? Human RBC or mammalian RBC. Fourth data I am writing about ostrich egg. And the next data I am writing about is nerve cell. So these are the size of the cell. Size is what? Variable. So what are the cell of the size of the mycoplasma? What NCERT wrote? Quote the NCERT data. NCERT wrote 0.1 or 0.3. How much? Mycoplasma. 0.3 micrometer. And what NCERT wrote about bacterial cell? 3 to 5 micrometer. What NCERT wrote about human RBC? 7 micrometer. What NCERT wrote about ostrich egg? It is largest cell. And nerve cell is one of the longest cell. Ostrich egg is that much big, it is visible to which one? Naked eye. Able to understand or not? So these are about the which one? Size of the cell mentioned in your textbook. Able to understand? So mycoplasma, student got a confusion and they came to us, sir, what to do in mycoplasma? In some place it is written 0.1 micrometer. So 0.1 micrometer is an example of mycoplasma. That is the smallest mycoplasma. That is which one? PPLO. PPLO. Getting the things or not? Is it? Pleuropneumonia-like organism. It is a smallest mycoplasma. Mycoplasma is a group of bacteria who don't have cell wall. Mycoplasma, if you talk about in mycoplasma, who is absent? Cell wall absent. So these mycoplasma is a group of bacteria, such bacteria who don't have cell wall, and many organisms came in that. So they just simply wrote a typical mycoplasma 0.3 micrometer, because in mycoplasma, like mammalia, many animal came na. How can you say about the size of a mammalia? Tiger have a different size, and elephant have a different size, human have a different size, cat have a different size. So they give a data like this, a generalized data. Mycoplasma 0.3. Mycoplasma is a group. It is not the name of a single bacteria. Are you understanding the idea or not? So they have given average size. If you talk about pleuropneumonia-like organism, that is which one? 0.1 micrometer. Is it okay? It is an example of mycoplasma. Is it done? Can we move ahead? Yes. Now we talk about shape of the cell. It is also variable. It depend on function performed by the cell. It depend on which one? Function performed by the cell. Shape depend on function performed by the cell. If you talk about some of example of cell, it may be cuboidal, it may be columnar, it may be polygonal, it may be irregular, etc. So what shape is there? An example I am telling, when you teach when you study the zoology, there sir told columnar epithelium, cuboidal epithelium. So cell type which is making a tissue, columnar epithelium have a different nature of function. Cuboidal epithelium have a different nature of functions. Are you getting the idea or not? So cell shape depend upon the function. Able to understand? Getting the idea? So it is associated with the function. So these generalized idea is over. Now we are going to deal about which one? Prokaryotic cell. Now we are going to discuss about prokaryotic cell. Is it okay? So prokaryotic cell, where it is present? Example, present in members of kingdom Monera. Kingdom Monera. Getting the idea? Prokaryotic cell. Example, you can say cyanobacteria. You can say about archaebacteria. Getting the things? You can say about cyanobacteria is a kind of which kind of bacteria? It is a type of eubacteria. Able to understand? You can say about mycoplasma. Everywhere prokaryotic cell is present. You can say about actinomycetes. You can say about chlamydia. You can say about rickettsia. Getting the idea? So these all are example of which one? Prokaryotic group. And out of that all prokaryotic group, these mycoplasma is the only group where cell wall absent. Able to understand? So you can say like, except mycoplasma, Cell wall present in every prokaryotic cell. Getting the idea or not? Right. So these are the example of prokaryotic cell. Archaebacteria, cyanobacteria, you can say about eubacteria, mycoplasma, actinomycetes. These actinomycetes is also called filamentous bacteria. Filamentous bacteria. Got the idea? Chlamydia, rickettsia, these all have which kind of cell? Prokaryotic cell. Now we are going to deal about the typical structure of a prokaryotic cell. We are going to deal about what basically? Typical structure of a prokaryotic cell. Can we move ahead? You have a look. So a normal bacterial cell, we are taking an example because bacterial cell are prokaryotic cell. So we are taking a normal bacterial cell as an example of a prokaryotic cell. So let me first draw the diagram, then we will just talk about the detailing of it. So you just have a look. This is the one layer, another layer I am drawing. This is the second layer, and then I am drawing the third layer of a typical bacterial cell. This is the third layer, and in third layer what we are finding? Infoldings are present. In third layer, infoldings are there. So if you talk about a typical most common, you can say prokaryotic cell, is trilamellar. Trilamellar. Trilamellar means what? It is covered by how many layers? 3 layers. 3 envelopes are there. You are getting the idea or not? Is it? So this is the outermost layer. We are saying it what? Glycocalyx. Outermost layer is glycocalyx. Second layer from outside is cell wall. And the innermost layer is cell membrane. Now, how examiner can torture you from here? Examiner can ask a question, which of the compulsory structure present in every bacterial cell? Out of these 3 layers, how many are compulsorily present? Plasma membrane. It is quite possible in some bacteria. That is the reason I told you, typical bacteria, not in all prokaryotic cell. In some one, who is absent? Glycocalyx is absent. It is quite possible a prokaryotic cell is there and glycocalyx is not there. Who is absent? Cell wall is absent in some. Right, like mycoplasma even don't have cell wall. So who is compulsorily present? Cell membrane. You can't find any cell where cell membrane is not there. But if you talk about a generalized thing, in all, not in all prokaryotic cell, in most of the prokaryotic cell, we find how many layers? 3 layers. Glycocalyx, cell wall, and which one? Cell membrane. So these 3 layers I drawn first. I will explain these layers later on as well. After that, here in plasma membrane, what you are finding? Infoldings are there. So here I am just writing the labeling of this infolding as what? Mesosome. And another infoldings I am just writing here as what? Chromatophore. Chromatophore. Chromatophore is not present in everyone. Present in only in which one? Photosynthetic bacteria. Present in which one? Photosynthetic bacteria. It is not a compulsory structure. Able to understand? Clear? Is it clear? Now, inside of the cell, I already told who is not there. Compartmentalization not there. So you will find who is present. Here, single circular DNA is present. And this is which one? Nucleoid. This is which one? Nucleoid. Now, in some, not in all even, in some prokaryotic cell, we are finding there is an extra circular DNA, plasmid is present. Who is present? Plasmid is present. So nucleoid is the main DNA. And plasmid is which one? Extra DNA. Who compulsorily present? Nucleoid. Whose presence is not compulsory? Plasmid. So plasmid not compulsorily present in every prokaryotic cell. Keep in the mind. But who must be present? Right? Clear? Now, here you will find who is present. Then it is cytoplasm, fluid-filled matrix. This is the cytoplasm, this is the fluid-filled matrix. In cytoplasm, what we are finding? Ribosomes are present, 70S ribosome. So these 70S ribosome, if many ribosome join together with mRNA, it is called which one? Polysome. Got the idea? If single free or ribosome may present, you can say ribosome may also present freely, and it also may present as a which one? Polysome. Able to understand or not? Now what we are finding? Who is present other than ribosome? Who is present? Just have a look. Who is present here? Inclusion bodies are present. So here I am just putting certain dots. Inclusion bodies. Suppose that these are different kinds of which one? Inclusion bodies. So these are what basically inclusion bodies are present. These inclusion bodies are present. Able to understand or not? Inclusion bodies are present. Right? Okay, ji. So now if we talk some more thing, some extensions of the cells are present. May or may not present. So these extension may be what? Flagella. Getting the things? These extension may be what? Fimbriae. So these extension are what basically? Fimbriae, and some another kind of extension may present. It is tubular in structure. It is called pili. Got the idea? Is it clear in the mind? Okay, ji? Right? So this is a structure of a typical prokaryotic cell. Now we are going to make it detailing. Right? So first of all, we are starting from which one? The outermost glycocalyx. Getting the things? Have a look here. So if glycocalyx, it is of two possible types. Glycocalyx made up of what? Chemical composition. Anybody can write? Glycocalyx chemically, what basically? Glycocalyx is glycoprotein basically. What is this? Glycoprotein. Glycans, proteins means oligosaccharide chain attached with which one? Protein. So if you talk about chemical nature of glycocalyx, in one line I am writing glycoprotein. Mostly glycoproteinaceous. So glycocalyx is two possible type. One is which one? If it is loose sheath like, if it is loose sheath, and if it is tough. When it is loose sheath, it is called what? Tell me. Slime layer. And if it is tough, it is called which one? Capsule. So the bacteria who have capsule is called capsulated bacteria, and who don't have capsule is called what? Non-capsulated bacteria. Getting the idea? Now if I ask a single liner question, what is the function of this glycocalyx? What function it is doing? Of course, if it is a boundary, first function is what? Protection. And second function is prevent bacteria from drying. Prevent bacteria from drying, getting dried. Means desiccation. Means what? Desiccation. Understanding the idea or not? So these are, if you talk about this slime layer, these are jelly-like layer. Able to understand or not? So it prevents it from getting dried. Able to understand? Understanding the idea or not? So you got the things about the glycocalyx. If it is loose sheath-like a structure, it is called slime layer. If it is hard, tough, it is called capsule. And the composition wise, what basically? Composition wise, it is glycoproteinaceous. Means with protein, carbohydrate chains are attached. Able to understand? Now I am telling about the second thing, cell wall. So space is not there, so I am writing cell wall here. Cell wall. So I wrote already, present in all prokaryotes except mycoplasma. Except mycoplasma. Are you getting the idea? Except mycoplasma, it is present in which one? Every prokaryotic cell. Now, what are important about cell wall? Cell wall composition. What are the composition? Composition of the cell wall. So it is made up of peptidoglycan. It is made up of which one? Peptidoglycan. And if you talk about peptidoglycan, it is made up of cross-linking of NAG and NAM in eubacteria. It is present, NAG and NAM present in which one? In eubacteria. NAG and NAM in eubacteria. Able to understand? NAG, N-acetyl glucosamine and N-acetyl muramic acid. Is it clear? N-acetyl glucosamine and N-acetyl muramic acid, right? So these NAG and NAM present in the bacterial cell wall. If you talk about function of the cell wall, protective. Protective. Getting the things. If I talk about the third thing, third layer, that is which one? Plasma membrane. Plasma membrane. So here a lot of things is there to say. If we talk about plasma membrane, so as you know, plasma membrane is made up of which one? Composition. Composition, phospholipid and protein plus protein. It is like eukaryotic plasma membrane. I'm just writing NCERT line, eukaryotic plasma membrane, because we will deal about plasma membrane in very detail na, so I'm not talking much about composition. Plasma membrane is a separate topic. Eukaryotic cell, it is like eukaryotic cell. Able to understand or not? Okay ji, understanding the idea? Now if you talk about cell membrane, of course thin, semi permeable, or better to write not semi, selectively permeable. Permeable. Here we are not talking about much about cell membrane. We are just going to talk now about infoldings in the cell membrane. Mesosome. It is what basically? It is infolding of cell membrane. It is infolding of cell membrane present as vesicle, tubules or lamina. Getting the things or not? Present at vesicle, tubules and lamella. Getting the things? It is infolding of the plasma membrane or cell membrane. So see in the diagram here, what basically? See here in the diagram. It is what basically? It is the infolding of the plasma membrane. And these infoldings are present as which one? Lamella, tubule or vesicle. Why it is not dragging? Yeah. It is mesosome. So what I wrote? These mesosome is infolding of what? Cell membrane, and they are present as what? What are the function of the mesosome? From here a question is going to ask. So can you tell me the function of the mesosome? Function of the mesosome? Yeah, I am expecting it is a long-term batch. I am expecting. Function of the mesosome. Very good. Cell wall formation. Cell wall formation. What another function you can say? DNA replication. DNA replication. And its distribution. In two cells, right now? Its distribution. Third function, cell secretion. Third function, cell secretion. Fourth function, it increase surface area for cellular respiration. For cellular respiration. Getting the idea? We can say it work like mitochondria of eukaryotic cell. So what job done by eukaryotic cell? In eukaryotic cell by mitochondria, so it work like mitochondria of eukaryotic cell. Getting the idea or not? What job done by mitochondria in eukaryotic cell? That work done by whom? Mesosome. Getting the idea or not? So what I told about mesosome, cell wall formation, DNA replication and its distribution in daughter cell, cell secretion, increase the surface area. For which purpose? Cellular respiration, means it work like what? Mitochondria of eukaryotic cell. So aerobic respiration can occur in prokaryotic cell or not? Yes, na? For aerobic respiration, what required? Mitochondria. But here who is absent? Mitochondria is absent. So who is working like mitochondria here? Who is working like mitochondria here? Mesosome working like mitochondria. Getting the idea? Now we are going to deal about which one? Another enfoldings. Another enfoldings, what? Chromatophore. So if you talk about chromatophore, it is present in photosynthetic. It is present in photosynthetic bacteria. So in chromatophore, who is present? It contains photosynthetic pigment. Photosynthetic pigment. So whoever the prokaryotic cell performing photosynthesis, their chromatophores are there. I drawn a enfolding here. Also, you can visualize in the diagram. Able to understand? I just make a enfolding in upper layer, and in that who is trapped? Pigments are trapped. Getting the idea? So this is what basically chromatophore. Now we are going to talk about which one? Nucleoid I already told, na? Nucleoid already told. Single, circular, double-stranded DNA. So no need to say it once again. Getting the things? Very good. You can say chromatophore as what? Work like chloroplast. Getting the thing. So now I am telling about plasmid. Already told about nucleoid in beginning, na? So I'm not wasting the time in that. Plasmid. So it is extra circular double-stranded DNA. And it is single also. Why I am writing single? Separate. It is single. NCERT wrote what? Single. Single means what? Not single-stranded. Many times student confused and came to us, sir, in NCERT it is written single circular. Single doesn't mean single-stranded. Single means present one. And that is the reason what? I have written it is single. It is double-stranded. Don't confuse. Many times student confuse here. Single as single-stranded. No. Able to understand? So, some plasmid, so the gene for, or better to write, it contain gene for antibiotic resistance. So some bacteria got the resistivity against antibiotic, na? They're not killed by which one? Antibiotic. So these antibiotic resistance gene is present where in the bacteria? Not in the main DNA. It is present in which one? These extra DNA. Able to understand? These extracircular DNA, plasmid. Are you getting the idea? About plasmid, we will talk much in biotechnology. So simply we are talking about extra double-stranded DNA. It is single. It is circular. Able to understand or not? Okay. Right. So NCERT wrote single means not single-stranded. And it may be more than one plasmid present in a cell. Is it or not? So have a look here. Now after plasmid, we are going to deal about which one? Ribosome. Ribosome. Ribosome is what? Associated with 70S. When many ribosome, prokaryotic cell we will finish today. Please be with me. Attached together with help of mRNA. It is called which one? Polysome. Polysome or polyribosome. Function, what function it is serving? Function is protein synthesis. Protein synthesis occur on ribosome. Function is which one? Protein synthesis. Getting the idea? Now we are going to say about which one? Inclusion bodies. What we are going to deal? Inclusion bodies. Inclusion bodies. If you talk about inclusion body, it is example I am writing Synophysian granule. These are storage granule. A storage granules. And it is non-membranous. Important. It is non-membranous. Got the idea? So Synophysian granule. You can say starch. It store what? Starch. Store starch. Glycogen granule. Clear from the name. Nobody will ask. It store glycogen. Store glycogen. Then you can say phosphate granule. Nobody will ask question. Clear from the name. Store phosphate. Who will ask in the paper? Which one? Synophysian granule. Store starch. These are important for. Another is gas vacuole. Gas vacuole is also a which one? Inclusion body. And it's present in which one? Photosynthetic form. Present in photosynthetic bacteria. Bacteria. And help in buoyancy. Buoyancy, to float on the upper part of the water. Many times a student came to us and asked, sir, you told these inclusion bodies is non-membranous. What about gas vacuole, sir? What about the gas vacuole, sir? Right, I think it is also your question. It is also your question. So here, these gas vacuole, vacuole is there, means vacuole have a membrane. But these are not a lipoproteinaceous membrane, not a true membrane. Getting the things or not? And that is the reason we are saying keeping it in inclusion body. We are keeping into inclusion. It is not a true phospholipid bilayer plasma membrane. Trilamellar, what you can say, plasma membrane. It is not that unit membrane, plasma membrane. Getting the things? So it is not a true membrane. So that is the reason here we are writing gas vacuole also in which one? Inclusion body. Now we are going to come on which one? Flagella. 3 more lines are there, the things will over. Now we are coming on flagella. So be with me patiently. Flagella. If you talk about flagella, 3 components are there in flagella. One is which one? Basal body. Another part is which one? Hook. And the third part is filament. So filament is the longest part. Filament is the longest part. Basal body attached with cell wall and cell membrane. Getting the things? Hook connect basal body with filament. With which one? Filament. If you talk about the structure is like this. Here you can say basal body, and then this part is what basically? Hook, and then this longer part is what basically? Filament. So this hook is joining the filament and which part? Basal body. This is hook, this is basal body, and this is filament. Got the idea? So flagella made up of flagellin protein. Flagellin protein. It is different from eukaryotic flagella. Eukaryotic flagella is made up of tubulin protein. It is made up of flagellin protein. Function of the flagella. Function, locomotion. Here now question not going to ask. Otherwise we taught rings in the flagella. How many rings are there? How many rings are there in gram positive? How many are there in gram negative? In cell wall I forget to say one thing. Based on the staining, that is technique developed by Christian Gram, right? There are two type of bacteria, gram positive bacteria and gram negative bacteria. Who get the gram stain is called gram positive, who not get the gram stain is called gram negative. We stain color with which one? Crystal violet. Crystal violet. Getting the things or not? Now function locomotion. Now after flagella, we are going to say about fimbriae. This is hair-like bristle present all over the body of bacteria. Present on all over the body. And these fimbriae have a role in which one? Attachment. Function? Attachment. And finally I am writing one line. Which one? Pili. It is tubular structure. NCERT wrote fimbriae and pili all together. They not write the different function. So tubular structure. And if you talk about these pili made up of pillin protein. Pillin protein. And if you talk about pili, it have a role in formation of conjugation tube. Conjugation tube during sexual reproduction. Able to understand or not? Right? So fimbriae have a role in attachment. Pili have a role in reproduction. Conjugation tube means sexual reproductions. Able to understand or not? Is it clear? So this is about a typical prokaryotic cell. This is about a typical prokaryotic cell. Got the idea? Is it clear in the mind? Right? So flagella, one very important thing. Flagella, in flagella who is not present? 9+2 arrangement absent. Absent in flagella. So how the flagella is there? It is uni-striated. It is uni-striated. 9+2 arrangement is absent in the prokaryotic flagella. That present in which one? Eukaryotic flagella. So you got some difference between prokaryotic flagella and eukaryotic flagella. So these are about the which cell? Typical prokaryotic cell. Is it okay, Ji? Any problem? Any doubt? You can say, ask. One question is there, up to when YouTube live continue? For few more days, probably. Right. Reason some students are coming late. So we just not find, they just lose the things. Still some people are coming. So we are just finding they not lose. Because whenever these topic lagging occur, so they will also just be in the communication with the topic. Whenever they want, they will read. And that is the intention. That is the purpose. Right. About, sorry to say, I just forget to say about books in courier. So I just now I will tell to the people who are looking this. Okay, what are the things? Okay, Ji. Thank you. Thank you very much. About the books I will tell. Okay. Okay, good. Welcome. This is the QR code. Weekend exam, this weekend, not but the next, now coming weekend exam is there. You will got the syllabus. Tomorrow we will announce it, right? Okay, bye-bye. Have a good day to all of you.